Water quality guidelines are currently provided by the EPA and are based on reference sites which are supposed to be streams which are in their natural condition. However, it's been recognised that many of these aren't and are in fact affected by human activities such as agriculture. And so we're trying to see if alternative methods could be used to help set these guidelines. So, water quality is important, as all these talks seem to address, as it affects biodiversity, stream-based recreation activities, commercial activities, and also the quality of drinking water. This is especially important here in South Australia due to the lack of water and also due to issues such as the algal blooms experienced on the Murray River. However, despite the importance of water quality, there are many difficulties in measuring it. So chemical spot sampling doesn't capture the full range of variability, and macroinvertebrate assessments are more affected by local habitat than water quality. So for example, this is one of the drains in the southeast that was sampled, and even changes in water quality here would have no effect, would not be picked up by the macroinvertebrate assessments, as basically it's so polluted that small change in water quality would still result in no decent macroinvertebrates being present there. So this is where we come on to diatoms. Diatoms are a class of aquatic algae and make their cell wall out of glass, and then these are some of the species of diatoms that we found in our samples. And they've been well established to be indicators of water quality with parameters, and are affected by parameters such as pH, nutrients, and salinity. They also incorporate chemical, biological, and physical water quality measurements. So we're interested to see if these could be a different indicator and a kind of more holistic indicator of water quality within South Australia. However, despite diatoms being well-established indicators of water quality, there's no standard method to sample them. So throughout this project, rope was used as the sampling substrate, as it was thought to be kind of the ideal substrate, as you could add a new clean rope into the water, it would be standardised, and the diatoms which would grow on the rope within 6 weeks would be the diatoms which were present in the stream within that 6 weeks. So this was why rope was used. However, it was also costly, as it required 2 field trips out, one to lay the ropes and one to pick them back up. So it also meant it was time consuming. So we wanted to see if the ropes were in fact actually the best way to sample diatoms, or whether natural substrates would have done the job just as well to save time and money. So these were our questions. We firstly wanted to find out if there were any significant relationships between diatoms and water quality, to understand how the diatom composition has changed along a nutrient gradient. Secondly, we wanted to see if there were any thresholds present which could be used to advise water quality guidelines. And thirdly, we wanted to see if the sampling substrates affected the study conclusions. So these were the sampling sites were identified by the EPA, and there were 30 sites identified in each area. The southeast was sampled in the spring and autumn of 2014, and the Mount Lofty Range was sampled in the spring and autumn of 2015. Although 30 sites were identified, not all were successfully sampled due to issues such as the ropes being lost or some streams being dry. At each site, the EPA also measured over 300 environmental variables, of which we chose a subset of 20 variables to try and cover a range of different environmental variables, and also ones which we thought would be important to assess changes in diatom composition. So, our method. We deployed 3 ropes at each site, and then they were left for 6 weeks before being collected. In the spring of 2015, mud and rock samples were also collected to test to see for this difference in substrates. As soon as the samples were collected, hydrogen peroxide was added methylated spirit was added for preservation, and then they were taken back to the lab. In the lab, hydrogen peroxide was added to remove organic content, and then the samples were washed 3 times before being put on cover slips and then mounted on slides using NAF racks. Around 300 valves were counted on each slide to the lowest taxonomic level, which was normally species level. And so, the 5 most abundant species of diatom found from each area showing here with increasing conductivity. So these are the sites here, and then these are the species of diatom. and the conductivity is here. It's worth noting that in the southeast, conductivity reached 20,000 Siemens per centimetre, whereas in the Mount Lofty ranges it only reached 6,000. These dotted lines are conductivity thresholds, which I'll get back to at the end of the presentation. This shows that Agnanthes minutissimum, Cochlinearis placentula, and Tabulera fasciculata were the most abundant species in both samples. And there are also similar patterns, with Agnanthidium minutissimum being present in streams which had lower conductivity and Tabulera fasciculata in streams which had higher conductivity. However, despite these general trends, there were differences between the two areas, with species of diatoms such as Planethidium delicaculum being the most abundant in the Mount Lofty ranges, but not even featuring in the top 5 in the southeast. So, to understand how the species composition changed along environmental gradients, we used canonical correspondence analysis. This is a constrained ordination technique that extracts the environmental variables which have the most explanatory power, and the length of the arrow shows how important the environmental variable is. So here in the Mount Lofty ranges, conductivity came out as the most important and has the longest arrow, so it was more important than like latitude, which had a shorter arrow. The sites here, the closer together the sites are plotted, the more similar they are, and the ones with circles are the samples from the autumn, and the ones with crosses are the samples from the spring. So yeah, in the Mount Lofty ranges, conductivity came out as the most important. And then in the southeast, conductivity again came out as the most important. Interestingly, we were expecting nutrients, or we were hoping that nutrients would come out as the most important explanatory powers, but TKM was only important in the southeast, and nutrients didn't feature at all in the Mount Lofty ranges. So, to further test the diatom's relationship with environmental variables and to see if this result with conductivity held true, we also looked at a diatom index. The diatom index we used was the Diatom Species Index for Australian rivers, and it uses sensitivity values assigned to individual diatom species to calculate an overall score, where high scores indicate a diatom sample from streams with better water quality than streams with lower values. We then tested the index scores against measured environmental factors to see if there was any correlation between the environmental variables and the diatom index score. So we found that there was a significant correlation between the index scores and conductivity in all data sets except for spring in the Mount Lofty ranges. We're still unsure as to what caused this one to not be significant, and so we're still looking for answers for that one. Longitude and percent covered by filamentous algae was significant in the autumn Mount Lofty ranges, but as these were not significant in any others, we wonder if this is more by chance than being causal. So, to understand if there were any significant change points in the diatom composition at specific levels of conductivity, so whether the change in diatom composition was a straight line or whether there were thresholds present, we calculated to see if there were any significant thresholds present using a method called threshold indicator taxa analysis, or TITAN for short. We first of all calculated the thresholds for the individual Mount Lofty ranges and southeast data separately, but we found out that this method was sensitive to small data sets, so we combined the two areas together to produce the overall thresholds. So we found that that there was a threshold both at 1,000 and 4,000 microsiemens per centimetre. The lower threshold is where species which prefer lower saline streams tend to drop out, and the higher threshold is where more saline tolerant species start being introduced into the diatom composition. We then tested this threshold using the diatom index, and we found that there was a significant difference in the diatom index above and below this higher threshold, and then it was 0.08 for above and below this the lower threshold. So finally, to get back to our rope issue, we collected samples from rock, mud, oh gosh, that's supposed to say rope, not cross, rock, mud and rope in the spring of Mount Lofty, in the Mount Lofty ranges. So each of the 3 samples from the same site are joined together by these lines to create the triangle. This plot here shows that there was that there was a greater difference between samples from different sites than from samples taken from the same site using different substrates. This tends to indicate that the different environmental variables between different sites had a greater influence on the diatom composition than the different substrates, than the different microhabitats from the different substrates had within the same site. We then also checked this again using the diatom index, and even to us the results came out as surprisingly similar and surprisingly nice, that there was no difference between any of the substrates. So we basically found that using rope, despite all its theoretical good points, was in fact a bit of a waste of time and money. We could have done it using natural substrates. So overall, in conclusion, we found that changes in diatom composition are primarily determined by conductivity and not nutrients as we previously expected. And we tentatively suggest that thresholds could be set at 1,000 and 4,000 microsiemens per centimetre, but we would like to test this on bigger data sets first to ensure its reliability. And then finally, the choice of substrates has minimal effect on results over an inter-site scale, so between our sites. So future sampling could use natural substrates instead of having to use ropes and go out twice. So finally, I'd just like to thank all these people, and especially Deborah, for sitting in the lab with me, helping me identify all the diatoms. So thanks.
Thanks, Jenny. Any questions? Fred?
It's a bit surprising that the turbidity has not any impact.
Yes.
Or any indicator for the underwater line.
So no, it didn't come out at all. And it's also interesting that even with the different substrates, when we looked at it, so the different with the rope was hanging in the water, the mud was at the bottom, and the rock could be up in the water, and it still didn't have effect on those diatom compositions from the different substrates across.
So the rope was sitting on the sediment?
No, it was ideally hanging in the water.
Oh,
so it was tied to trees so that it was dangling in the water flow. But of course,
the profile.
Yeah, but of course, because we sampled over spring and autumn, as the levels went up or down, it could affect where it was in the stream.
Any other questions? Got time for another one? Yeah?
What are the implications of the study in terms of freshwater aquatic biodiversity?
In recording biodiversity?
Yeah.
So we would argue, the streams with the higher water quality would have higher biodiversity. So we were thinking that having a better diatom community or having more diverse diatom community could be representative of streams with high biodiversity. So kind of using as a proxy between the two. Is that what you meant?
Yeah.
Okay.
Okay. One more question, if anyone has one.
One of the Southeast, I can't remember which one, showed that TKN was
That's
second significant. What do you think that's TKN not really acting as a nutrient? So that's not an oxidized nitrogen. So any idea what that means?
No, we're interested in finding, with further stuff that I haven't put up here, is that when you compare the rope and take out the conductivity, so use partial correspondence analysis, some variables can vary together, we're getting an undercurrent of nutrients might play a slight role in it, but no, I'm not sure what it is. But also things like max current, we've had some random interesting things come out which we haven't yet explained.